Just checking. All right. So when we're looking at mitosis, I want to remind you about some things. First of all, in the mitosis steps that I've given you that you need to know for the test is that there's this part that says pro-metaphase. Now, in the chapter reading, they give you all these little subsections. You do not have to remember that for mitosis. You need to know, for mitosis on the test I'm going to give you, you need to know, hold on a second, I'm trying to switch colors. So you need to know these steps, 1, 2, 3, 4, 5, and what is happening in each step. So you should know by now that the DNA is condensing in prophase, the nuclear envelope is going away, and you see how they combine prometaphase and prophase. So in the prophase that we discussed, the DNA goes through condensation, and you get 2 centrosomes, but the microtubules are not connected yet. The nuclear envelope disappears. In prometaphase, the microtubules connect, and they start to move towards the center. So they combine prometaphase and prophase into one. In metaphase, they're all lined up. In anaphase, you are separating the sister chromatids. Right? The sister chromatids are separate. You see them here? One sister chromatid here, and the other here. One key thing about and this is really important for metaphase, they are all exact copies. When I say that, I mean the cells. When you're at the end and you see here cell A and you have here cell B, both of these cells are exact genetic copies. They have exactly the same DNA. You have 46 chromosomes here, and then we say 2N. Remember what we talked about, 2N and N. We say 2N because there's 2 copies, 2 homologous chromosomes for each of the chromosomes that particular species has. So again, to be clear, with humans we have 23 different chromosomes, but we have 2 pairs. We have a pair of each, one from mom and one from dad. So we have 46 in humans. I'm trying to remind you that different species have different numbers of chromosomes. So what's key here is that there's 2 key things, is that these 2 cells are exact copies of one another. That's what you want for mitosis. When you start growing, you don't want each of your cells to be have different genetic information. That'll be very confusing. So that's mitosis. What makes meiosis just slightly more difficult, is that at the end you're not going to have genetic. In fact, there's kind of 2 overpowering reasons to do meiosis. Let me start off with this. If you wanted to have babies with your spouse and you had 46 chromosomes in your cell, and your spouse took one of his cells or her cells and took 46 chromosomes, and you combined them, how many chromosomes would your child have? If you took a regular cell, and you combined it with another regular cell from your spouse, you'd have 92. That's not right. We don't, because a human has 46. So this is wrong. This is not what we do, right? So we have to do a process, and we call that meiosis. And in that process, we do what we're basically doing, just basically now. I say this basically because there are some important differences. So you're going to have to know the important differences for the test. So you have the key here is that we have to take your 46, and we're going to copy them. We're going to do S phase. You're going to do S phase in the full cell cycle. And you're going to, when you have 46, after S phase, how many chromatids do you have? If you have 46 chromosomes, after S phase, how many chromatids? 92. So you have 92 copies of the chromosomes. Does that make sense? Because you had this thing that was a chromosome, and then it went through S phase, and now all of a sudden you have this thing here. So there's 2 copies. Now these are exact copies. They're sister chromatids. There's another word that people are confused about, and those are, I tell you that you have one chromosome here from mom, right? And you have, let me see if I can make this happen with blue here. And you have one chromosome here from dad, chromosome number 1, right? These are what are called homologous chromosomes. All right, so these chromosomes have exactly the same gene, and they have the same genes, right? So they have this gene for eye, and they have this gene for height. Does that make sense? Remember that? Because we went over this before. Do you understand that? They're both chromosome number 1. So all the genes here are the same. What's the difference between mom or dad? Well, no, yes, that's the big difference. That's one chromosome where they're not the same. But mom and dad, what's the difference in these? These are the same genes, but they have different what? Different messages, what we call those different messages, different versions of the genes, alleles, that's right. So they have different alleles. But because they have the same genes, we call them homologous. Is that clear? Now, so go ahead.
So that's the X thing? I don't know.
What do you mean X? You're trying to go right to the paper. What are you asking? Why are you flipping through the work? I'm asking you to listen to this. Okay, go ahead and ask your question that I misunderstand. Oh, this here? Are you talking about this here? What I'm telling you is that it's the same thing I've been telling you for the longest time, so it's very frustrating for me. But I'm going to try to say it one more time. In humans, you have 23 chromosomes. And you have 23 chromosomes. I'm not redoing that. You have 23 chromosomes, different chromosomes. Are we clear with that?
No.
All right, so 23 of them, and I'm not going to draw 23, so I don't know how to do this other than to tell you that there's 23 chromosomes of these. Different ones. Each one is different. Are we clear? So one chromosome will have eyes, the other one will have liver, another one will have kidneys. Are we clear? So each chromosome has different genes that make you who you are. Yes, the genes, the information on the DNA. The genes, yes, the switches. So each switch is going to make a different thing. So on chromosome number 1, you have your eyes. How do you make eyes? It's the information's on chromosome number 1. And you have one set that came from dad. This is dad's chromosome number 1. There's 23 of them that came from dad. Are we clear? Yes? Maybe? Sort of? Silently standing there? Come on, people. Someone get motivated here.
I got you.
23 chromosomes from dad. Mom has 23 chromosomes that she gave you.
Right, to make it 46.
Together you have 46. Why is it? What's going on? Are we understanding this? Okay. Now, we're going to go through S phase. You're in S phase. What's going to happen? This one, instead of having this one chromosome with one molecule of DNA, what do we do? What did S phase do? What does S phase do? A lot of homework. You have a lot of homework. Synthesizing is correct. Make copies is correct. Right? You're synthesizing DNA. You're copying DNA. So you have a copy. What are you connecting the 2 copies with? Centromere. Excellent. So you got the 2 copies. They're there. They're connected with a centromere. They are these sister chromatids. are exact copies. They both came from Dad, both of them. Because this one was copied, and it's here. This is a copy. Do you understand? Everybody understand that?
Yeah.
What don't you understand about that?
Like the thing that the central here, like who's are those connected together? That's what I'm
Did you study your DNA replication? I'm asking you a question. I need to know.
Yes, I studied it.
So you know what DNA replication is. You know the steps.
Yes.
So wherever you had an A, you put a T. You made exact copies. You have two molecules. I'm going to refer you back to that DNA replication video. And what this is this chromosome was copied, and that's what this is. I don't know what other words to use, other than the words that this one is copied and it's attached by a centromere. Help me out. Somebody help me out to say what magical word is going to get this in your head. Help me. Right?
Wait, what's she doing?
This, I don't know, I'm not sure where the confusion is. This is a chromosome that you were given by your dad. Chromosome number 1. You went through S phase. You copied it. You literally made a copy of it. It's sitting here, and it's attached by a centromere.
I think get it or not? Yeah.
I don't know what I'm saying wrong, so you have to help me.
Look.
Help me. I don't understand what I'm saying wrong.
I'm about to do something. I think I just walked.
You have this chromosome, and then you attach it when you make a copy to another chromosome. These are sister chromatids. That's what I'm asking. Do you understand that?
So you go through S-phase and it sits out the DNA replication. That's how you get the second copy. And then you make DNA replication. There's your daddy chromosome. S-phase. You make a copy. So it's the whole thing. Then you got the little friggly lines that come to the chromosome.
The microtubules and tear them apart.
To make the...
Do you remember that?
To make the back chromosome.
Do you remember that? It's over here. Let's go back over here. It's right here. There they are.
It's your daddy.
Look at it.
It's your daddy.
These are the sister chromatids attached. And then the microtubules separate them. One from mom. 2 copies of mom's, 2 copies of dad's, all 23. Pairs. We understand that? Okay, now you got it. All right. I don't know what I was saying wrong. They made a mistake.
You need some illustrate, some actual.
All right, whatever. So I need a piece of paper that I rip in half, but okay. All right, so anyways, do we understand this so far?
Yeah.
Okay, so now that's great for mitosis. That's right. We want exact copies. But meiosis, we don't want exact copies. And the key is that we need to take these 2 cells, your cell that you have 46 of, and your husband's cells or wife's cell that has 46. If you want to make a baby, any 2 diploid organisms that want to make babies, in other words, any 2 organisms that have 2 copies of each gene that want to make babies, you have to do something interesting. You have to split that DNA in half. Now, because you went through S phase, now you're going to have in here, when I separate them, you have 23 chromosomes, but how many copies of the DNA?
Put 24.
Oh, I don't know why I did that. So I want to confuse you, I guess. So we made copies. Remember, there's 2 sister chromatids, right? Each one's a copy of the chromosome. So if I have 23 here, I didn't separate the chromatids, but I did separate the homologs. Do you remember what a homolog is? Okay, so there are 2 copies of the chromosomes. I didn't separate the chromatids, so how many copies of DNA do I have here? No, I have 23 chromosomes. How many copies of the DNA? 46, because every chromosome has 2 chromatids. So there's 46 chromatids here. We need cells that have only 1 copy of each chromosome. In order to make a new baby, we need 23 single molecules of DNA, 23 different ones for humans. In other words, you need an N number for humans. N equals 23. 2N is 46, which is where we are right now. That's every cell in your body except your eggs if you're a lady, and sperm if you're a gentleman. So we're 23 each. We have to split them one more time because we have to split the chromatids. That's why there's 2 steps to meiosis. The first step, now we have 23 here's 23, but 46 copies. Right? And here's down 23, but how many copies? 23 copies. And hopefully you'll understand a little better in a minute. So now when we do this, of course, we do this both here for both cells, right? So at the end of meiosis, you're going to have four cells. You have 4 cells, and they're all going to be 23 chromosomes. Single copies of chromosomes. And mom, if this was dad, then mom would do the same thing. And at the end, she'll have 4 eggs. All of them with 23 single versions, not doubles. Not 46 copies, 23 copies. Now you can take one of these 23s and you get to combine it with another 23 to equal what?
46.
To equal a new human being with 23 pairs. 23 pairs, one of each from mom, one of each from dad, or 46 total. And that's how you were made, and that's how you're going to make a baby. Now that's the general outline. There's a lot of specifics. This is why I told you to watch that video, read the chapter, do the homework. But if you're not going to do that, you're not going to understand. Sitting here and understanding now is not going to help you on the test, because you're going to forget. You have to watch the video. You have to read the chapter. You have to do the questions. In other words, you have to do your homework. All right. All right, so when we take a look at, there's 2 phases, right, 2 parts. In prophase, in meiosis 1, we're starting with how many? 2n, 46. 23 pairs, 2 times 23, 46. We're starting with 46. Now, why do we put 2n? I want to remind you that every species has their own number of DNA. In humans, it's 46 pairs, or 23 pairs, 46. For adults, 2n equals 46. So that's why I'm using 46 here. But every species has a different number. On the end of course exam, I could see them telling you corn using 103.
Corn like food?
Yeah, your corn. Let's say it's 103 chromosomes. What's N? How many are going to be in each of its gametes?
You mean divided by 2?
Divided by 2. That's how many you have. It's always going to be an even number. All right. So, aside from that, what's happening in prophase 1? Well, we know what happens is the chromosomes condense, but then we got this weird thing that's called synapsis and crossing over. So these are the differences between mitosis and meiosis, okay? In mitosis, the chromosomes lined up this way. You had one there, that was chromosome number 1. Chromosome number 1 from dad was here. Chromosome number 2. Look at how they're lining up.
Right.
Along the metaphase plate. Remember that, the metaphase plate?
Yeah.
Now, what happened was the spindle fibers, as Mogaji so elegantly explained a minute ago, come off of the kinetic core, or which is in the centromere, right? And it's going to pull those 2 apart. Do you remember that?
Yes.
Okay, so that was mitosis. That's not what's happening here, because that wouldn't help us. That wouldn't help us because we'd end up with 46 chromosomes in both the cells. We don't want that. Everybody agree?
Yes. Mm-hmm.
I'm going to use 2 colors. I'm going to use pink for mom and blue for dad.
All right.
So each one in meiosis, what happens is you get this is after S phase, so you've made your copies. So you see the sister chromatids here?
Yep. Yeah.
Okay, these are sister chromatids. There's 2 copies in each chromosome. So dad is over here. Now, what do you notice is different about how I'm lining up now rather than what we lined up earlier? Go ahead, Latifa.
And this is called meiosis, right? And what mitosis, they don't line up like that, like.
They line up how?
Like they line straight, but
Straight
these
up and
are
down.
already next together.
They're together. And we call this process, it's different, it's called synapsis. It happens during prophase. You have to know when it happens. It happens during prophase. Yes?
Okay, I have a question. I've seen how you do the mom like real small and they do the dad bigger.
Yeah, I didn't mean to do that.
Oh.
That was not intentional. All the chromosomes are the same size because they all have the same general sequence, right? They have all the same genes. The difference between mom and dad is just the order, right? The alleles, right? We're all good with that?
Yeah.
Everything here is the same except for the alleles. They have just slightly different versions. Big A versus little a, right? Big B versus little b. See, mom would have little a, dad would have big A, mom would have big B, dad would have little b. You know what I'm saying? So they're different in that sense. All right. Is that clear to everybody what's going on here? But here's an interesting point. When they cross over, something weird happens. They exchange DNA. Oh, you knew that? Good. Sorry about that. They're exchanging DNA. Okay? So when let me redraw it. Let me show you what I mean. We're going to have to, I'm hoping that you understand that big A and little a are
The switch.
right. So let me go ahead and switch the DNA here. I'm hoping you understand that big A and little a are different versions of the gene. Same gene. I did it again.
Just different people, different versions.
Right. Same gene, just slightly different. And the other one is going to be something like, this is after crossing over, right? You see how they crossed over?
Wait.
Hold on, give me a second.
I feel like we got the same question.
So now, I changed the shape, don't worry about the shape, but yeah, go ahead.
Which one?
Go ahead.
Okay, is that conservation, like how you see is one strand of pink, one strand of blue, like the
Right,
DNA?
this is not semi-conservative, right? Semi-conservative was replicating the DNA. Different process, right?
Oh.
They happened during S phase. What's happening here is that you had this DNA molecule, they touched, and they switched. They exchanged it. Exactly at the same point, which I don't know if I did well expressing that here. So what I'm saying is that in this case, if originally you had big A, big B, little c, little d, both of the pinks had that sequence. Do you understand that? Mm-hmm. And Dad gave you little a, little b, big C, and big D. That's what Dad gave you. So the full-on pink is big A, big B, little c, little d. So blue eyes, dark skin, curly hair, tall. The Dad gave you brown eyes, light skin, straight hair, short. I can't remember what I said the first. Tall.
Yeah.
All right. So they look different. The genes that you give this is what you have. You ended up being whatever the dominant form of each of those genes are, but now you're giving it to your child. You're making sperm or you're making eggs, so you're going to give it to your child. So if you got mom's original DNA, what your child would get, if your child got that, your child would get from you. Who knows what your husband or wife would give them, but they would have this sequence. If you gave them this mixed chromosome, you see, this mixed one would be Dad's little a. This mixed one would be Dad's little b, right? And this mixed one would be Mom's little c. Still, see, it's still pink, and Mom's little d. So now you have, instead of big A, big B, big little c, little d, you have little a, little b, little c, little d. Completely different combination, right? Now we do this again, and with Mom you'd have big A, with still the pink you have big B, with Dad's blue you'd have little c, and with Mom's pink you'd have little d. So now you even have a third different combination, don't you?
Yeah.
Does that make sense? Do you see that? Do you see that? Do you see what I'm saying? I mean, don't just say yeah, because we're running out. We're done with time.
So,
We're on Tuesday already, guys.
so everything.
So with the blue, we have if the egg gets the full blue, you have little a, little b, little c, or is that big C and big D? So a fourth combination of these genes. So let me put this aside, and we'll come back to it in a minute. I'll just keep that in mind. Yeah.
So that means no combination will be the same?
That's right.
Alright.
So now instead of remember what we had before. You got to remember what we had before. What we had before was we had one possibility was this, another possibility was this, right? They were connected.
Right.
And then another possibility was this, another possibility this, and they were connected. So you only had really two sequences to go for, right? To go with. You either had Dad's version or Mom's version. Now that we mixed it up, now you got four. You got Mom's original version, Dad's original version, you got the mixed up one here or the mixed up one here. All of them have different combinations, don't they?
Yes.
So you have four different combinations. We call this genetic variation. Mark it, write it down. You're increasing, that's the one really big reason why sex is a better way of doing cellular reproduction, is because the way by recombining the genes, we also call it recombination. Crossing over is the same as recombination. And that makes sense, right? Recombination means you're mixing it up. So you're getting a more mixed up genes, more mixed up alleles, more mixed up traits. So now instead of having everyone in the family tall, you could get somebody who's short, right? You have a bunch of tall brothers and sisters, have one short one. Now you can have a bunch of dark-skinned brothers and sisters, have one light-skinned person. Now you can have a bunch of brown-eyed people, have a light-skinned person. Because now you have these different combinations. I said skin,
Yes,
I meant...
brown eyes.
I don't know, man, whatever. I confuse myself. Brown eyes and blue eyes, right? So you have all brown... You ever notice a family, everybody's brown eyes, you got one kid who's blue-eyed?
Yeah.
And people are like, what's going on?
Yeah.
It's this genetic variation. Yeah.
So with that, this is the reason why, like, worms and them their kids have the same type of, what's the name, enzyme, because they don't have that type of variation like humans do.
Yes, that's right. So you have to think about this. Nobody in here looks exactly like their mother. No one in here looks exactly like their father. Nobody. You might resemble them. You must resemble them, right? They gave you your genes. So inevitably, you're going to have characteristics that will remind people of your father or mother. You have characteristics of both. But nobody here looks exact. And oddly, the more important one is nobody here has a brother or sister who's exactly alike. Right. Even brothers and sisters who had the same parents. Obviously, twins are an exception, for God's sakes. Yeah, I knew somebody was going to say it, because we do have a pair of twins in here. But those are they're different. That's different. I wasn't going to say nothing until you all started talking.
Yeah.
So, anyways, brothers and sisters generally do not look exactly alike.
Right.
Twins are a different situation we're not going to even talk about. I don't even want to get into it. Yeah.
So if you were a boy and a girl, y'all both twins, would y'all still look alike?
See?
Not.
Did I just say? I said no twin.
She a twin. She got a twin too. I just wanted to know.
If you're a boy and girl?
Yeah, I got a boy.
See, this is why I don't want to get into twins, because it's a whole nother subject. We'll get into it later, but not now. It's going to get confusing. Twins are different situations. Fraternal twins, which is what you are. You are a fraternal twin, which means you were a fertilized zygote, and then you went through mitosis. Well, no, not you. That would be different. You had two...
See, that's why he don't want to get into it.
Oh my God. Yes, I don't want to get into it because it's difficult to explain. But your egg, before your egg was fertilized, that egg split, or your mom released two eggs, one of the two. And what happens is two different sperm connected those two different eggs. So you two are basically a brother and a sister that share the womb. The twins, you share the womb so you're born at the same time, so you're twins, right? But you're no closer genetically than any brother and sister are.
Right.
Now the Smiths are another situation. The Smiths were a fertilized egg, so they were zygote.
That's split too.
Then they split into two and they separated. They didn't like each other, whatever, they separated. And now these two people have exactly the same DNA. Became full human beings, and they're sitting at our desk right now. So these two people share exactly the same DNA initially. As we talked about earlier, they will change throughout time. They will not stay the same. Their DNA will change over time. But the sequence will never change. They'll always have A, B, C, D. The same A, B, C, D. The difference is which ones are turned off and on, which genes are turned off and on. That difference will change throughout their lifetime.
So how people born conjoined?
See, this is what I'm saying. This is where we get lost, and then you go home trying to figure this out, upset that you don't understand meiosis.
I get that.
All right, so moving on. In prophase 1, ladies and gentlemen, are there people asleep or texting in class? Because when your eyes are not up here, reaching into your book bag and texting is not writing and taking notes. I love how she looks around. You know it's you. I'm not going to mention your name. Don't mention. You'll say it out loud because I don't want you on tape. It looks suspicious. Your hands in your bag instead of writing and taking notes. That's all I'm saying. All right. So if I'm wrong and you're not texting, that's fine. I don't want to put anybody make anyone uncomfortable. I just want you to just do the right thing. All right, so prophase 1, okay? This whole thing we just called synapse, I just described to you, happens. So when we come out of prophase, now all 4 chromatids are different. Do we understand that? All right, so let's move on. I'm going to quickly do this, so hopefully it works. So in prophase, we go through that, we line up, but now instead of lining up the way we used to, right? Now we have these 4 chromatids that are lined up in what we call a tetrad. All right, you need to know what a tetrad is. That's necessary. A tetrad. Tetrad means there's 4 of them. There's 4 of them. That's right. Somebody's done their homework. So the tetrad. The tetrads are all connected, right? There's 4 of them. Notice there's 1 chromosome here, 2 chromatids, right? One that's original, the other one that's recombined, right? Here's another chromosome from dad one's original, the other one's recombined. Now they're going to line up here. Do you see them lining up here? And instead of separating the chromatids in metaphase 1, we separate the homologous chromosomes, right? We separate these chromosomes. So what we end up with is two going, this chromosome going this way, this chromosome going that way. In the end, during telophase, please pay attention and know this. When we get to telophase in the end, you're going to have 2 cells, right? There's going to be 23 chromosomes in each, right?
Yeah.
But each chromosome is going to have what? Is going to have 2 chromatids. Do you agree? I don't hear anyone agreeing, so I'm not sure what's going on.
Yes, I agree.
Okay, thank you. So you have 2 sister chromatids, and you'll notice that one sister chromatid is original that you got from mom. Then there's your recombined DNA that you made new. This is a new one. You made it by recombining you mixed up mom and dad's genes, alleles. Does that make sense? So now the same thing's going to happen in the other one, except the other one you got dad's this version, right?
And then a new one.
And you got a new one. That's right. You got a new one that has a little bit of pink and then some blue and then some pink. So these chromosomes then, this is your new one, okay? Here, and then here's your original. Now, what's even more interesting, just to mix things up a little more, because that's not enough genetic variation for life. Life wants to add even more genetic variation. That's where we get so many different possible combinations. By the way, I think the reason we're falling asleep is like hotter than blazes in here.
Don't do it.
It's 90 degrees, dude.
It's alright. It's cold in school.
Now, how come you're like this?
Because it feels good.
Because it's almost hours a week.
All right. So anyways, so we got the two chromosomes, they have 23, but that's not enough, because if we combine these two, we have 46 copies of DNA in the new cell, and we can't do that. So we're going to do this whole process again. Now pay attention now. Pay attention to what I'm saying. If you don't understand this, you can't do this packet. That's why that's why I wanted you to read the chapter. That's why I wanted you to watch the video. If you don't understand everything I'm talking about in here, that packet will not make sense. If you understand what I'm talking about, that packet will be easy. That's your choice that's in front of you. So that is the end of meiosis 1. In meiosis 2, our objective is to separate these. Now, our objective in meiosis 1 was to first recombine them to get some more increased genetic variation. In other words, do synapses, do crossing over, mix them up, and then half the DNA. So I halved it. So I went from 92 to 46. Wait a minute, Mr. Mendoza. You said 23. Yeah, it's 23 chromosomes, but how many copies of the DNA? 46, because there's 2 chromatids for each one. There's one more added genetic variation. Which one did you get? Did you get in this cell when you recombined, when you separated those chromosomes, right? When you see this, when you're doing this, and you're asking yourself, well, notice the blue one with a little bit of pink, so dad's chromosome and little mom's came over here for chromosome 1, right? Mom's chromosome with mostly red and a little blue went over here to this cell. Do you see that?
Yeah.
But in chromosome 2, it's the opposite. Mom's came over here with a little bit of dad's, and dad's came over here with a little bit of mom's. So this is also increasing genetic variation. This is random. So it's another step in increasing genetic variation as to which version. Do you get mostly moms or mostly dads? So the version of each chromosome is random. Does that make sense? So you just don't know what you're getting at the end is the point, right?
That's like another reason why you can't get the same combination.
I love you. That's exactly right. Exactly right. Brilliant. Absolutely brilliant. That's another reason why, she said, that's another reason why you can't get that same combination when you have a baby. You're not going to get, the odds of getting 2 people like the Smiths that are genetically identical, right? Without it being like a single egg, a single fertilized egg dividing, the odds of just doing this process and getting 2 cells that are genetically identical are infinitesimal, negligible, almost zero.
People can mutate it, like...
It's, I'm not telling you it's impossible. I said almost zero. It's just hard to believe it could even happen. So, okay. So 23 chromosomes. Got it? Now we got to do the second division. The only reason you do the second one is to separate the chromatids. So the second mitosis, second prophase, or second meiosis 2, do you see it here, meiosis 2, is exactly like mitosis. There's no difference. You're making an exact copy, and you're separating the chromatids. The only difference is you're not starting with 46 because you started with 23. You didn't do S again, did you? No. And you already separated the chromosomes. So when we go through the process for each of these, we end up with 4 of these. Pay attention now. This is where it gets interesting. Well, it's been interesting for me all along. But here you have 4 cells. You end up with 4 cells. And each one is going to be genetically different. And let me show you what I mean. I'm going to draw out the chromosome from this combination here, I'm going to draw out what each one of these eggs got. So in one of these eggs got exactly mom's version, original version that you got from your mother. Exactly the same. So there it is. Another version of this, another egg got dad's chromosome number 1. Now remember, this happened 23 times. And another egg got a little bit of dad's and mostly mom's and a little more dad's, right? And another egg got mostly mom's, a little bit of mom's, mostly dad's, and a little more mom's, right? So what you got in the end is that this one turned out to be big A, big B, little c, little d, whatever those characteristics are, right? Tall, short, brown-eyed, whatever it is. And somebody else got big A, big B, little c, little d. And then somebody else got little a, little b, they got what? I think little c, and they got little d. All recessive, right?
That's the same DNA. We thought it was supposed to be little a, little b, and big C, and big D.
Okay, let's go back and make sure I'm not doing something wrong. So when I look here, I see 4 different versions, right? Mm-hmm. I see little a, little b, little c, little d on this one. That's the one that ended up here, you see?
No, for the father one, for the original one, the second.
Yeah,
Oh, you're right. Yeah, I messed that up. You're absolutely right. Good catch, yeah. Oh, please. I'm gonna scratch it out. Is that all right? Little big A, this shouldn't have been this at all. This should have been, I'm gonna go ahead and do it, little a, little b, big C, and big D. Sorry about that. That's absolutely right. And then the last cell here, you got mostly dad. You got big A, big B, little C.
Big D?
I think it's little d. Yeah, little d. All right. So there it is. So now you got 4 cells. Now, if you can just imagine this happening 23 times, you can understand, I think, that what you're dealing with is a process that gets you 4 very different eggs. These 4 eggs are going to be very different. That's why you're so much different from your siblings. And then you can imagine that this happens in your husband's sperm as well. So he has 4 different sperms from his mother and father, from their mother and father, etc., forever, back to the original, right? And so here you have this really super different set of combinations. Which one of the eggs get fertilized? I don't know. Which sperm of the million sperm that your husband releases to fertilize? We don't know. So then when they get together and they fertilize, they become this absolutely unique being, right? You are a unique human being. There's never been anyone like you, and probably will never be anyone like you again. All right? You're incredibly, the odds of someone being exactly like you are just infinitesimal. Okay? So you're a unique human being, and that's it. That's meiosis. Now you'll notice that we went down to what? 1 N. So what are the key points to take home? You have 4. You started off with 1 that had a 2 N, right? You whittle it down to 1 N each. And remember, and take a look at all the different colors, each of those ends is going to be different. So you have a different combination, each of these. You see that?
I thought that as a question.
See that again?
Okay, like, so the blue was supposed to be for dad and the pink was our mom. So the 2 that switch, like, they gonna stay the 2 that switch, and then the originals just stay the original ones?
That's right.
Them just all switch?
That's it. That's exactly right. Not all 4 switch, right? So that. What does that do? That if all 4 switch, we would get a lot of different. Maybe we'd get more combination, maybe not. But this is just the way it happens. Only the chromatids that actually touch, and you see here the ones that touch. Only the 2 that touch switch.
So what keeps the 2 what keeps the 2 original ones from not switching?
They don't touch. It's how they're lined up. So if they touch, they'd switch. All right, but they don't touch because of how they're lined up in the cell during the process of metaphase. Do we understand this?
Yeah.
All right, so try to answer what you can in the last couple minutes. Look at them. Look at all the questions. Ask me a question. Ask me a question that's confusing in that I'll check the work tomorrow. Obviously, I don't have time. Look, a lot of people have been disappointed, but there's no time. What do you want? I can't.
With this.
I will put it all in the grade book tomorrow when we're going over these packets, mitosis and meiosis.
Okay.
Right now, look at that packet, and this is your opportunity to ask me a question without having to come after school. This is Tuesday, so I can't stay after school today. Go ahead.
Why do you have
Number 2. What is this? Let's look at number 2. Quickly, let's all look at number 2, since that's the difficult one.
Okay.
What would happen if human sperm and egg were produced by mitosis? What would happen? Tell me. You'd have 92 chromosomes, and it wouldn't work, would you? Because if you did mitosis, you started with 46, you'd end up with 46. They're exact copies. And then you try to combine the two, sperm and egg, what'd you get? 92. I think you could have figured that one out. I don't think that was that hard. I think you could have got that. Look at those questions and give me something that's hard. Don't ask me the easy ones. You're killing yourselves. Don't forget to do the mitosis stuff. If you can answer these questions easily, this is your check. This is how you check to see if you're ready for the test. If you can answer these easily, you're pretty much going to get an A in that mitosis section of the test. All right? Because this is the hard. These questions are the level of difficulty, your end of course exam for mitosis. This is the level of difficulty for the questions on your final. So if you can't answer these, you need to see me tomorrow after school. All right, so I'm done recording. You got about a minute left. Work until the